Edwardsiellosis. Edwardsiellosis is one of the most important bacterial diseases in fish. The common agents in Edwardsiellosis are Edwardsiella tarda and Edwardsiella ictaluri. Edwardsiellosis has zoonotic potential, which means this disease can also infect humans. The hosts in Edwardsiellosis includes various marine and freshwater fish, tilapia, Pangasius, hypothalamus, and eel. Edwardsiellosis is distributed worldwide. For clinical signs, in Edwardsiella tarda, the affected fish will look lethargic, hang at the surface of water, and swim in spiraling or erratic pattern. The gross external lesions vary with species, such as small cutaneous ulcerations, which normally seen in channel catfish, swollen abdomen, which normally seen in flounder and cichlid, ulcers on the head, which normally seen in bream, and hemorrhage on the body and fins, which normally seen in common carp, Japanese eel, and striped bass. Common external lesions includes abdominal distension, rectal hernia, absophthalmia, and hyperemia. Internally, the most common gross lesion consists of light-colored nodules on the kidneys, spleen, or liver. Meanwhile, for Edwardsiella ictaluri, the affected fish will refuse feed, tend to hang at the surface of the water, and swim with a spiral movement that includes erratic bursts. For gross external lesions may include hemorrhage around the mouth, body, lateral and ventral portions, and fins, pale gills, absophthalmia, and small ulcerations on the body, or always referred as hole in the head disease. Internally, petechia occur or develop throughout the visceral mass and in the peritoneum and body musculature. Some fish may develop ascites, and the liver, kidneys, and spleen are commonly enlarged. This figure shows external signs of olive flounder infected with Edwardsiella tarda, which includes abdominal distension and rectal hernia, absophthalmia and opacity of the eye, and also peripheral hyperemia in mandible lesion. This figure shows Japanese flounder juvenile that was naturally infected by Edwardsiella tarda, where the abdomen is distended and shows prolapsed rectum and large intestine. A large amount of serum sanguineous ascitic fluid accumulated in the abdominal cavity, and the liver is severely pale and mottled with multiple abscesses, and it is also covered by a febrile hemorrhagic exudate. The transmission in Edwardsiellosis may include fecal-oral route, cannibalism, and direct contact. For diagnosis, cultivation can be made on standard medium at 22 to 26 degrees Celsius. Definitive diagnosis is based on standard biochemical tests and agglutinations. Other than that, fluorescence antibody detection, PCR, LAMP, loop-mediated isothermal amplification method, and ELISA can be done. For treatment and preventive measure, antibiotic treatment based on sensitivity tests accompanied by improvement of water quality can be done. Other than that, stress reduction, housing, hiding places, population density, and group composition in the tank can be done, and strict quarantine procedures and stress prevention can also help to prevent this disease. There are available vaccines in Edwardsiellosis such as formalin-killed cells, FKC, LPS, ECP, live attenuated Edwardsiella tarda, avirulent Edwardsiella tarda, ghost cells, OMP, recombinant proteins, recombinant protein expressing cells, OMV, and also the NA vaccines. There are several predisposing factors in Edwardsiellosis, such as intensive fish farming methods, overuse of antimicrobial chemicals in aquaculture, bacterium's broad host range, and also development of multi-antibiotics resistance. That's all from me. Thank you.